All right. Good day, everyone. Again, my name is Dorothy. I am a cell molecular and developmental biology major, mentored by Dr. Monica J. Carson of the Division of Biomedical Sciences. And today I am presenting on sex-specific inflammatory gene responses to LPS systemic inflammation in the murine jejunum. So, inflammation is currently a focus of research in many fields because it's a prominent feature of many diseases, accounting for over 50% of deaths and disability in the past years. So, if you think about it, it's good to have the immune system. It's really cool. But the cost of having it is that it will always react to things that are unfamiliar to it because it is trying to protect us. And this leads to the development of inflammation, which sometimes becomes a bridge to a more serious condition. So here I outlined some inflammation-related diseases and the average number of people affected by them. So we have fatty liver, diabetes, neurodegenerative conditions, but of course several others. I work in a neuroscience lab, and so we are going to take the brain, for example, because it is central to memory and bodily functions. So it turns out that brain inflammation can actually start outside its area, like from gut, for example. And our lab is interested in understanding the gut and brain interactions because of how this could affect our brain. So you might be wondering, what's the sex-specific part all about? So very few labs actually have defined the differences in male and female inflammatory responses because of the cost of doing so. And so females often lack representation in the samples, which leads to one-sided evidences. But because of certain factors that make each sex unique, it is equally important to understand their differences in immune or inflammatory responses so we could better determine potential treatments for conditions that occur in both sexes. And that is why we are looking into that. Now, in our body, inflammatory responses can either be local or systemic, with local being limited to only one organ or area of the body, while systemic, which is what we are more concerned about, is more global, involving multiple organs and sometimes the whole body. So we want to study sex-specific differences better in the lab and how our body reacts to systemic inflammation. So we mimicked human inflammation using LPS, or lipopolysaccharide, being applied to mice. And this is a part of a gram-negative bacteria. You might have seen it in my title. And it triggers immune response without the damage caused by a real bacteria. So first we inject LPS into the animal's abdominal cavity, and then this triggers a local immune response in that area. But eventually, the inflammatory signals released by the cells around that area will travel into the bloodstream and cause what we so call the systemic inflammation. So in this event, different tissues would react to these signals, and other people in our lab focus on the brain, but my project focuses mainly on the part of the small intestine called the jejunum. So like any part of the intestines, the jejunum is lined with epithelial cells connected by a junction protein called claudin 3, which are represented by the green squiggly squiggles here in the bottom figures. And these junction proteins, claudin 3, act like Velcro for the cells to keep them together tightly so nothing is leaking out. And we found that during inflammation, these claudin 3 are internalized, and this is indicating local tissue response, and you can see that in the bottom right figure. So we decided to investigate them previously, and I want you to remember those green proteins because they're the same proteins that we see in here. They're also green, and so the ones that are encircled in red are individual cells of the jejunum. So when claudin 3 are internalized, we are going to see tiny green dots in the center of the cells like we would see on the top right image. So if we look at the leftmost images, those are our controls. We're not supposed to see the green dots in the center of the cells. In the bottom right, the males that are treated have a couple of them, but surprisingly, the females are showing us a lot of these internalizations. And so we counted these dots or claudin 3s for all our samples and found that yes, these LPS treated females do have more claudin 3 internalized, and this is telling us that females are responding more robustly to the inflammation compared to the males, at least in the tissue. So we looked at the changes in the expression of inflammatory genes as well, and these figures are called volcano plots from our nanostring data, and these measure the materials or inflammation-related materials that the cells are making. And as you can see on the left figure, there are more genes that are represented by dots that deviate from the center, which is showing that females are having more changes in the expression of their inflammatory genes compared to the males. And this is consistent with the previous slide. So we took those genes from the previous slide and put them in these Venn diagrams to better see the changes in what is happening in these genes. The main thing that caught our attention is the upregulation of CCL2, which is a signaling molecule. So CCL2 attracts macrophages into the inflammation site. So these are types of immune cells. And we thought that this might be interesting because it is only upregulated in the treated females. So we might observe greater number of macrophages in the treated females' tissue compared to the males. So just to illustrate my point on how these CCL2 molecules are attracting the macrophages, here we have our healthy and inflamed jejunum. In our healthy jejunum, the macrophages migrate to the tissue just for normal turnover, and this always happens because they're usually just in the tissue and they need to get replaced at some point. But in the inflamed, the jejunum's tissue is releasing these molecules called CCL2 represented in blue, and they are supposed to attract more macrophages. So more macrophages should be recruited. And since these molecules are only upregulated in females, we predict that females will have greater number of macrophages in the inflamed site or inflamed group compared to the males. So to answer this question, I have outlined our experimental design in here from left to right. First, we inject the abdominal cavity of our treated groups with LPS, and then we collect and prepared samples and labeled a unique protein in the macrophages so that we could distinguish it from other cells. And then we took pictures and counted the cells using the software called ImageJ and compared our 4 groups. So here I have the representative images for our 4 groups, and we could see the macrophages as the tiny white dots that I've encircled in red. And if you look at it, the LPS females, which is the bottom right, and LPS male actually do have greater amount of these white dots. But when we did a counting, that was actually not the case. So we counted the macrophages using our software, and again, we expected females to have greater number of macrophages, not the males. And our data shows that yes, macrophages on the right is not increased in males. But unfortunately, if we look at the left figure, it is also not increased in females, which is really interesting to see. And so, while we saw sex-specific differences in tissue response earlier, the green ones, and the upregulation of the chemical signals that attract macrophages, like the ones in the Venn diagrams, we did not really see an increase in macrophage cell count in either the males or the females. But the thing is that doesn't really tell us the state of these macrophages, because I find the shapes of these cells interesting, and their shapes can determine their activation. So part of our future direction is that we are going to analyze the macrophage morphology or shape, because they could tell us the state of these cells. And we just started doing this using the ferret's diameter in our software. And then I will come back next time when we do have new findings. So I would like to thank Carson Lab, especially Dr. Carson, who is here right now, for her support and helping me develop my skills, and our grad student, Paula, for performing the experiments in person on campus and for additional guidance. And of course, the Mark U Star program and my fellow trainees. And that's all for me. Thank you.
Thank you for that presentation. We have a couple of minutes now